When we talk about ruminants, we usually think of our domestic ruminants, cattle, sheep or goats. You will have in mind the picture of grazing cattle and know that a short time after eating, the animals lie down and begin to ruminate. The animals masticate about 50 seconds for each bolus. The remasticated material is swallowed, and after a short break, the next bolus is returned to the mouth for further mastication. These events can be seen not only in cattle, but also in many other animals which belong to the great group of ruminants. This goat was fitted with a rumen fistula in order to elucidate the motor function of the forestomachs. In this drawing, the internal structures of the reticulorumen are shown. Both forestomachs form a large fermentation room. The reticulum is divided by the reticuloruminal fold from the cranial sac of the rumen. The cranial pillar, longitudinal pillars, and the caudal pillar separate the rumen into a dorsal sac and a ventral sac with a caudoventral blind sac. The cardia enters the forestomachs in the upper part of the reticulum. The reticular groove runs from the cardia to the reticulo-omasal orifice located in the ventral part of the reticulum. In order to record motility of the reticulorumen, open-tip catheters are introduced from the rumen fistula into the reticulum, into the cranial sac, into the dorsal sac, into the ventral sac, and into the caudoventral blind sac of the rumen. The goat is fixed in a special stand. The catheters are connected to electronic pressure transducers. Behind the animal, you can see the necessary carrier amplifiers and a multi-channel recorder. From motility records, the sequence of contractions of the various compartments can be seen. The reticuloruminal cycle starts with a diphasic or double contraction of the reticulum. Then contractions of the cranial sac, of the dorsal sac, of the ventral sac, and of the caudoventral blind sac follow. Now a second contraction of the dorsal sac occurs, followed by a second contraction of the ventral sac of the rumen. After a short break, the next reticuloruminal cycle starts again with a double contraction of the reticulum. The cranial sac of the rumen is maximally contracted when the reticulum has already been relaxed. The dorsal sac of the rumen is contracted maximally when the cranial sac has already been relaxed. The contraction of the ventral sac starts after relaxation of the dorsal sac, and the contraction of the caudoventral blind sac starts after relaxation of the ventral sac. Again, secondary contractions of the dorsal sac and of the ventral sac of the rumen follow. First of all, the sequence of the reticuloruminal cycle is shown in a schematic drawing. A swallowed bolus has passed from the esophagus into the reticulum. During the double contraction of the reticulum, the lumen is reduced and the fluid contents flow over the reticuloruminal fold into the relaxing cranial sac of the rumen. At the same time, swallowed food is forced rearwards. During relaxation of the reticulum, the cranial sac contracts. By this means, the fluid contents of the cranial sac flow back into the reticulum over the reticuloruminal fold, whilst light solid particles are forced over the cranial pillar into the dorsal sac of the rumen. During the following contraction of the dorsal sac, the solid contents are squeezed out and are slowly rotated. At the same time, the pillars of the rumen move dorsally and are constricted to a small ring. Thereby, the contents of the middle part of the rumen are forced ventrally. During the following contraction of the ventral sac of the rumen, the ring-shaped pillars move ventrally. Thus, the fluid contents of the ventral sac are mixed and small amounts pass into the cranial sac over the cranial pillar. The contraction of the caudoventral blind sac follows. The gas bubble is therewith expelled into the dorsal sac, and the pulpy contents of the caudoventral blind sac are compressed. During the following second contraction of the dorsal sac, the gas bubble is forced into the cardia so that eructation can occur. The second contraction of the ventral sac of the rumen has the same effect on the contents as the first. In order to look directly into the forestomachs, the rumen fistula will be opened and the contents of the dorsal sac removed. The forestomachs were illuminated with a light cable which was fixed on a retractor and introduced through the fistula. Now we are looking directly through the fistula into the forestomachs. Here you see the upper part of the cranial sac of the rumen and at the bottom, the reticulum. By the double reticular contractions, the digesta are expelled into the cranial sac. On the right side, the cardia and the upper part of the reticular groove can be seen. Another contraction of the reticulum. First contraction, second contraction, and now backflow. In the foreground, the curved reticulo-ruminal fold is visible. Now the goat is fed with grass. When a swallowed bolus passes into the reticulum, a rapid protrusion of the esophageal mucosa between the lips of the reticular groove occurs. In the empty stomach, the boli are forced into the cranial sac beyond the reticulo-ruminal fold. By the reticular contraction, the boli are slowly pushed over the cranial pillar into the dorsal sac of the rumen. When the fluid digesta return into the relaxing reticulum, solid particles are kept in the cranial sac by the reticulo-ruminal fold. Below, one can again see the cranial pillar of the rumen. In order to look into the ventral part of the reticulum with an endoscope, the forestomachs are totally emptied. The bucket with a volume of 7 litres is filled up to the top. Now we are looking into the forestomachs, which are filled with saline solution. We are facing from the cranial sac to the crescent-shaped reticulo-ruminal fold, and beyond the fold you see the reticular cells of the upper part of the reticulum. Some grains of seed, which were introduced into the reticulum, are thrown from the bottom of the reticulum beyond the reticulo-ruminal fold by the contractions of the reticulum. Now we introduce the endoscope into the reticulum and are facing towards the bottom of the reticulum containing the settled grains of seed. The red balloon is filled with lead for better relaxation of the reticulum. During the reticular contraction, the bottom of the reticulum is lifted and the grains of seed are thrown to the reticulo-omasal orifice on the right. After relaxation, the particles with higher specific gravity rapidly settle to the bottom of the reticulum and are kept in the reticular cells. During the reticular contractions, the reticular cells are compressed and the settled particles are emptied owing to the very quick movement. These events can be followed up several times. Here you see a reticular cell in close-up. The small papillae might be of use for keeping macerated food particles in the reticular cells during relaxation. Now the reticulum in radioscopy. The clip on the top is fixed at the reticuloruminal fold. By the double contraction of the reticulum, the digesta are expelled over the reticuloruminal fold into the cranial sac of the rumen, and afterwards they return into the relaxing reticulum. Another reticular contraction. During the successive contractions of the cranial sac, the ventral pole of the cranial sac is lifted up to the level of the reticuloruminal fold. 2 table tennis balls filled with barium sulfate are introduced into the reticulum on the left and into the cranial sac on the right. By the reticular contractions, the light ball is thrown into the cranial sac of the rumen. It flows, together with the fluid contents, back into the reticulum. This sequence can be followed once more. First phase, second phase of the reticular contraction. Gas from the dorsal sac of the rumen passes together with the backflow of digesta into the reticulum so that eructation can occur. After a reticular contraction, gas is forced again from the dorsal sac of the rumen to the cardia. The second clip on the right is fixed to the cranial pillar of the rumen. After the reticular contractions, the cranial pillar, right clip, moves in dorsal direction and, during the contraction of the ventral sac of the rumen, in ventral direction. At this moment, digesta may pass from the ventral sac into the cranial sac of the rumen. We're now facing the dorsal sac of the rumen. The bright area beneath the spine is the dorsal gas cap. The mass of solid food is dark. During the contraction of the dorsal sac of the rumen, the gas cap disappears and the rumen wall contracts down onto the ingesta. The mass of solid ingesta is squeezed by the compression of the wall and the pillars of the rumen. During relaxation, the gas cap returns. Another contraction of the dorsal sac of the rumen. Because the cranial parts of the rumen are attached dorsally to the abdominal wall, the dorsal sac of the rumen moves in cranial direction during contractions. The contents are slowly rotated. Now we are facing the dorsal sac and the caudoventral blind sac of the rumen. A contraction of the caudoventral blind sac is immediately followed by a contraction of the dorsal sac of the rumen. By the compression of the caudoventral blind sac, gas escapes into the dorsal sac. It returns during the successive contraction of the dorsal sac and relaxation of the caudoventral blind sac. In the next scene, the alternating contractions of the caudoventral blind sac and of the dorsal sac of the rumen can be seen once more. Contraction of the caudoventral blind sac, gas escaping into the dorsal sac. Contraction of the dorsal sac, gas returning into the relaxing caudoventral blind sac. Now the goat is fed, and one can afterwards see the passage of the digesta through the esophagus. Boli arrive at the cardia, are retained for a moment, and then forced into the reticulum. During eructation, gas passes from the reticulum into the esophagus. Now we are facing the upper part of the reticulum and the cranial sac of the rumen. A swallowed food bolus is ejaculated from the esophagus into the reticulum. It is forced rearwards by the contractions of the reticulum. Another bolus is swallowed. It is also forced rearwards by the reticular contractions. The food boli are now in the area of the cranial pillar of the rumen. They are slowly displaced over the cranial pillar into the dorsal sac of the rumen. The goat continues eating food. The swallowed boli are successively moved into the dorsal rumen. Now we follow the movement of the food particles through the rumen. By the contractions of the pillars, the food particles are displaced from the dorsal sac downwards into the caudoventral blind sac, which can now be seen. In the following scene, we will observe the events of regurgitation and rumination. In sheep and goats, a regurgitated bolus is remasticated for about 45 seconds and then swallowed. After a short interval, the next bolus is returned into the mouth and continuously remasticated. The regurgitation, which occurs at regular intervals, is related to forestomach motility. During a period of rumination, the contractions of the reticulum and rumen are recorded again. At the end of the reticuloruminal cycle, a separate regurgitation contraction of the reticulum occurs. At the maximum of this contraction, reticular contents are aspirated into the esophagus. Now, the regurgitation contraction is followed by the normal diphasic contraction of the reticulum. Whilst the digesta are remasticated, the reticuloruminal cycle proceeds in the sequence already described. Before the reticuloruminal cycle is finished, remastication is stopped and the masticated bolus is swallowed. After a short break, another bolus is returned to the mouth at the beginning of the next reticuloruminal cycle. Now we are again looking directly into the reticulum. Although the forestomachs are partly emptied, regurgitation occurs. The goat tries to aspirate digesta into the esophagus. Here one can see that digesta are aspirated at the maximum of the reticular contraction. Because light coarse material is carried by the reticular contractions back into the dorsal rumen, digesta from the lower part of the reticulum are aspirated, which will predominantly consist of fluid containing small food particles. Now we are facing the thoracic region. Gas has passed into the esophagus, and now digesta are aspirated for remastication. The fluid contents of the bolus are swallowed. Now deglutition of the remasticated bolus and the next regurgitation. Now we are facing the throat. The goat swallows the remasticated material. After a short interval, the next bolus is returned to the mouth and the fluid contents of the regurgitated bolus are immediately forced back into the forestomach by double redeglutitions. Again, back to the thoracic esophagus. Redeglutition of the remasticated bolus. Regurgitation. First deglutition, second deglutition. These events are repeated with surprising regularity. From this observation, it can be concluded that the digesta are returned to the mouth very quickly and that the fluid contents of the regurgitated bolus are swallowed immediately by two deglutitions. Let us sum up in which way food particles move through the reticulorumen. They pass into the reticulum and are forced back into the dorsal sac of the rumen by the contractions of the reticulum and of the cranial sac. Here the food is squeezed and slowly rotated. Smaller particles tend to descend into the ventral rumen, and by the contraction of the ventral sac they are returned to the cranial sac, and from here back into the reticulum. Small particles are forced from the reticulum into the omasum, whilst large particles of the reticular contents are ruminated. The remasticated digester pass again into the reticulum.